Good afternoon.
Welcome, and the floor is yours. Thank you.
Thank you. Thank you, Joanna. Hello, everyone. My name is Ken Okaji. I am an independent coral reef consultant hired by the Okinawa Prefecture Government as a research coordinator. First of all, I'd like to thank Mr. Halton King and the organizing committee for giving this opportunity. Today, I will talk about some results of the Okinawa 7-year research program on Cots outbreaks. Excuse me. The major threat to reefs in Okinawa is the crown-of-thorns starfish in the past 5 decades. Degradation of reefs is not only an important environmental issue, but also a serious economic problem for the region. These local fishermen and tourist operators have been struggling with Cots removal by hand for many, many years. There was a criticism that Okinawa government should prioritize research, but there are no human resources. Also, reefs were so close to the life of local people, they couldn't just sit and watch their corals get eaten. This figure shows the number of Cots removed from reefs in various areas between 1973 and 2003. The first control program was conducted in 1969. The local government bought out removed starfish from local fishermen. The price was about 20 to 90 cents per Cot. Such bounty system was introduced from Micronesia because Okinawa was under US administration at the time. Then the control program was switched to subsidy in 1979. And in the next 3 decades, hundreds of million yen was spent to remove millions of Cots, but most reefs were devastated. Without an effective strategy, either the bounty nor the subsidy could not concentrate the control efforts in the manageable area. Consequently, many Cots aggregations remained on reefs in 1970s and 1980s, and these aggregations reproduced secondary aggregations, so-called chronic situation. And based on these past failures, the Okinawa government has adopted a revised control policy. The current control program is conducted only in a limited, manageable area that local people want to protect. And conducting scientific research was another decision that the Okinawa government made in 2012. This figure shows rainfall and the timing of Cots outbreaks. These bars indicate 3 months rainfall, May, June, and July, in which Cots spawning takes place. These dots indicate the primary and secondary outbreaks. Line is the length of chronic period. As I mentioned, the first primary outbreak was in 1969. A small aggregation appeared in the west coast of Okinawa, the south of Onna village. Then the big aggregation spread to north in 1972. 3 years before these outbreaks, 1966 and 1969, there were heavy rainfall. The second primary outbreak was in 1996. At the time, Cots aggregations were comprised of 2-year-old individuals. Looking at this pattern of rainfall and Cots outbreaks, we thought that the nutrient runoff hypothesis might also stand in Okinawa, like as a high islands in the Pacific. So we had a total of 17 research projects under 3 major themes: water quality, larva nutrition, and larva disposal. I guess some of you had been to Okinawa for 2004 ICRS. Okinawa main island is located 26.5 degrees north and 128 degrees east. These islands are surrounded by fringing reefs, not like the Great Barrier Reef. The climate of Okinawa is monsoonal, hot and humid in the wet season, May, June, July. The seawater temperature in summer months ranges between 28 and 30 degrees Celsius. Unfortunately, we did not have severe coral bleaching in recent years. So from 2013 to 2018, we surveyed concentrations of chlorophyll A and neutroids in summer at 13 sites, about 60-kilometer distance along the west coast of Okinawa main island. The southern coast is a populated area, including the capital city, Naha, and the northern coast includes the outbreak initiation area. The result, the 5-year overall average chlorophyll was 0.2 microgram per liter, with a yearly fluctuation between 0.15 and 0.3. The chlorophyll A along the southern coast was much higher than those of the northern coast. When we looked at the seasonal fluctuation, chlorophyll was higher in summer, particularly for the southern coast, the red line, and chlorophyll in the southern coast in summer stayed in the range between 0.25 and 0.8 that cod larvae are supposed to survive and grow. In contrast, chlorophyll in the northern coast, the green line, was near or lower than 0.25. And these figures show the daily changes in nutrients, chlorophyll, and POMs. The survey was conducted near the northern coast from July 6 and July 19, 2014. This figure shows ammonia, phosphate, nitrate and nitrite, and chlorophyll, total chlorophyll, and the phytoplankton chlorophyll, and flagellates and bacteria. This blue bar indicates the day of big rain from a typhoon. As you see, nutrients quickly went up right after the rainfall. The next bacteria, the flagellates, increased the next day, and chlorophyll increased in 2 days after. Every river and gullies in Okinawa are short and very steep, so that Okinawa fringing reefs are strongly influenced by terrestrial runoff. And this figure shows the relationship between chlorophyll and survival or growth of Cots larvae. According to these data, Cots larvae are starved when chlorophyll level is below 0.25 microgram per liter, which is normally observed in the northern coast of Okinawa. The chlorophyll survival curves were drawn by Dr. Katarina Fabricius based on my larva rearing experiments at Lizard Island back in 1993. Actually, my today's talk is the first talk at an official meeting in Australia after I left Townsville 25 years ago. So at this opportunity, I would like to express my sincere appreciation to these organizations and people who supported my study in Australia. The main focus of my PhD study was to identify the major diet of Cots larvae. From old days, Cots larvae was thought to eat phytoplankton in the size range between 2 to 60 micrometer in diameter. The lower limit, 2 micron, is the distance between cilium and cilium. And the upper limit, 60 micrometer, is the size of the Cots larvae slot. So based on the direct microscopic observation of their gut contents, I reported that Cots larvae hardly feed on phytoplankton, such as cyanobacteria and bacteria. So with the recent technology, we use stable isotope analysis to see if Cots larvae can utilize particulate foods other than phytoplankton. It is known that carbon stable isotopic ratio of predators corresponds to that of prey with about 1 per mil isotopic enrichment. The nitrogen isotopic ratio of predators generally indicates larger enrichment, about 3 per mil. So in this figure, red circle indicate the changes of carbon and nitrogen isotopic ratio of Cots larvae bred in natural seawater. And the blue circle are for Cots larvae bred with only microalgae. So on day 0, Cots embryo's carbon and nitrogen isotopic ratio was similar to that of adult starfish, which is almost the same as coral. And the ratio of starved larvae showed only slight change. However, the ratios of these fed larvae gradually decreased in 10 days. And finally, the ratios of larvae reared in natural seawater were significantly lower than those of the larvae reared with microalgae. And these results suggested that Cots larvae assimilated natural particles other than phytoplankton because a large proportion of organic carbon in natural seawater was consistently comprised of phytoplankton and detritus. And with the different experiments, we also confirmed using radioisotope that Cots larvae were able to assimilate coral mucus. Surrounded by a large amount of organic particles, food limitation in Cots larvae may not be severe as long as they stay in the coastal waters. So next, larval dispersal. We released a total of 25 GPS buoys from the initiation area on a village during the spawning season in 2013 and 2014. These figures show the tracks of GPS buoys in 7 days after release. Several buoys were washed up on the northern beaches, but the other went up north along the Kuroshio Current. These are the tracks of buoys in 30 days after release. A few buoys went offshore, but then came back to the vicinity of Okinawa Island. And interestingly, one buoy went further north and reached Japan mainland in 1 month. But the question was, if larvae are drifted away to offshore, do they survive? So one of our team members simulated Cots larval dispersal using a statistical model. He used the actual ocean current data and chlorophyll data, an average 3 weeks of larval period, according to the chlorophyll survival response curve was also incorporated into the model. The model was run 10,000 times, assuming that 10,000 larvae were released from 30 different areas. So let's see an example of simulation. Release area was set to 4, Philippine, Yaeyama, Miyako, and Kerama. Cots larvae spread out to wide areas, but there is a limit because chlorophyll A is generally lower in offshore waters. So in most simulation, a large proportion of larvae did not travel long distance. 84% larvae released from Yaeyama and 47% of larvae released from Kerama did not go beyond 50-kilometer circle. And the proportion of larvae traveled beyond 500 kilometers was less than 0.1%. This is another result of simulation. Most Cots larvae released from the initiation area on the village returned to the same area. So I mentioned that the first record of outbreak in Ona was in 1969, but many local fishermen had noticed gradual increase of adult starfish from the mid-1960s. As Dr. Yasuda presented, there were no outbreak records not only from Okinawa, but also nearby upstream regions such as Taiwan and Philippines since the early 1950s. So the past record seems to support our result. If this simulation is close to the reality, Cots larval dispersal leading to successful recruitment is thought to be a local scale event. And if that is the case, water quality improvement for coastal waters should be an important challenge for the Okinawa government. Finally, on behalf of the Okinawa government, I would like to thank to Dr. Peter Doherty, Jamie Oliver, Sylvain Mutike, British Africa, and every scientist involved in Cots research at the Australian Institute of Marine Science for their helpful and generous support to our research program. Thank you very much.